So I'm going to sort of hopefully motivate some ideas. Two main things I'm going to be looking at today is when you have data, how you build statistical models to actually find out something that you want to know. So the whole point of collecting data is not just to collect data for the fun of it, it's to learn about the system that you're interested in. So it's thinking about how we relate the system we're interested in and want to learn about to the actual data that we have, and from the data what we can learn from the system. And in order to do a statistical analysis and do this, I've sort of split it into two parts. One of them is how you actually build a statistical model, basically what links the real world with what you see. And the second part is how you actually fit the model you've now built to the data to actually learn things about what you want to. And I'll do some case studies. I'll do as they're running through things. So please feel free to ask questions as we're going through. It's sort of not going to go too in depth in any particular one example. So already said, we're interested in some system. Climate change, probably one of the most topical ones with regard to the environment. You know, what has the climate changed over time? In which case you need to collect data of the climate over time, and then that you want to be able to answer the question, is the Earth getting hotter? Is this abnormal? What's causing it? And so on. But more general than that, environment's changing. Deforestation is another example I'll touch on ever so slightly later. But in all these problems, all these things that we're interested in, the whole point is, how do we learn about the system? We go and collect data. So first thing you have to do is, so what data should I go and collect? You need to collect data in a sensible way such that you are able to answer the question. So I'm supposed to be like, I'm straight in front of your slides, aren't I? You need to be able to answer the question that you're asking. If you want to know, in London, has the climate changed in the last 100 years? There is no point just looking at weather variables at different cities around the UK, because you're looking at spatial information, and what you want to know is temporal information. However, you might be interested to know, how does the weather change around the UK, in which case it would be sensible to be looking at spatial information about weather variables around the UK, rather than just looking in London over time. Both types of data are sensible, but you have to think about what question you want to answer in order to know which data you go and collect in the first place. So you should know what you're interested in before you just go and randomly collect data. Unfortunately, in some cases, people collect data and then think about the question later, and the data they've collected is not able then to answer the question they've realised they want to ask. So we should think about these things from the start. So, different types of data. I've sort of split them up with regard to environment and ecology. Spatial data, so where data is collected at different points in space. Temporal data, we collect data over time. Spatial temporal data is where we have spatial and temporal data. And individual level data is really more looking at, well, in my way I'm focusing on ecological type things. So where you collect data on an individual, typically over time. So it's a much lower level type of data rather than the larger sort of environmental type data. So your data may look, we'll give examples of each of these different types of data. I've realised that looked a lot, okay, perhaps it makes the point, okay. So this is just spatial data. It's where you have data that's basically like a snapshot at a time. So if you look at the one on the right, that's just a point sort of pattern, and it's to do with the location of particular species of animals in a particular species of plant in an area, and it's a marked point process in the sense that we've got two different types of species, and we might be interested in how they interact in terms of how the plants grow. Do they cluster together, or are they inhibiting each other, and they have to be spread out? Whereas the data on the left is satellite data of somewhere in Africa. I can't narrow it more than that. And it's looking at the composition of what's on the ground. So in particular, they want to be looking at different patterns, so where there might be deforestation, or you can see roads appearing and so on. So they want to be able to categorise what's happening at the ground level, where you can't actually go there. You have to look at it from satellite data. So that's just sort of spatial data. And And it can be lots of other different forms as well. So again, weather covariates, weather variables around the UK, it's sort of fixed locations. So temporal data, simply data over time. So the left-hand side is estimated count of an index for the number of lapwings at certain sites around the UK. And you can see the interesting things are happening, and in particular there appears to be a big crash in their population size, and then you've got that probably is observation error. We'll come back to that probably later. But you can, from that we want to know what's happening about the population over time and explain what's happening there. Whereas the one on the right is to do with rainfall in some way, I think in the US, I can't remember now where I pulled it from. The rainfall, the different lines are for different years. So each line, each colour line is for a different year, and it's looking at a month, and you might be looking at what's happening. And you can see, hopefully, some sort of trend that they all seem to go down between January and February, and then there's some sort of possibly seasonal effect going on there. And then we can combine these together to get the spatial or temporal, where we have the spatial patterns over multiple time points. So again, the top one, we've already seen this for just one snapshot, but we might have multiple snapshots, and it's to do with the satellite data over time. Similarly, the bottom plot, so again, these are our locations of muskoxen at different times, and this is in an area, and the colour code is to do with the altitude. So now you've got information at the spatial scale and temporal scale. And the last type of data we might often meet is individual level data. So it's at an individual level, and in our case, think of individual animal. So the plot on the left, there is actually some output, and the blue, the dots, orange, grey dots are to do with the location of an animal. So there's a GPS tracker on the animal, and they record the location at certain intervals. And then the rest of it is to do with the model that we fit to that data. Data on the left, for those of you who were here this morning as well, is capture-recapture data, where we go out into the field at a series of capture occasions. First time we go out, all the animals that we see we round up and we uniquely mark them and then let them go again. And we uniquely mark them. It means if we ever see them again, we know it's that same animal we've seen before because they put a ring on its leg or a tag in its ear with a unique ID marker. And you go out again at the second time point, do the same thing, round up all the animals, you record if you've seen them or not, and so on. And because we've uniquely ID'd the animals, we have that each row is the capture history for each individual animal that we see within the study. A 0 means we didn't see it, a 1 means that we saw it alive, and a 2 means we recovered it dead. And again, we'll come back to that type of data later. And of all these different things, we can ask different questions and hopefully answer them from the data that we have. So we've gone out, we've collected our data, and now we want to use that data to learn about the system from which we've observed this data. And in order to do this, we need to link what's really happening, what the true state of things are, with the data that we observe. Because we can't typically observe everything in the, sort of a population level, we only observe a sample. So our data is simply a sample from what's really going on. And it's from that sample we want to learn something about the real system that there is. So in order to do that, we define a statistical model that basically describes what's happening at the system, and then how that system, what's happening there, relates to the data that we observe. So again, there's these two types of processes going on, what's really there, and then the observation process. What do we observe given what's really there? And then we have a statistical model that we set up. It's a function of things that are of interest to us, so model parameters that are sort of the things that we're interested in, which is why we collected the data in the first place. And then we want to estimate those model parameters from the data. And typically we estimate them and have some level of uncertainty associated with them. So that's sort of an introduction into the general idea of data, statistics, and particularly in the environment and ecology. So the first part I'll mention, so we've got two parts. First part is how do we build these statistical models, and then the second part is, given the statistical model, how do we fit them to estimate the parameters that we're interested in. So I've split them up into two parts. This talk, and we'll see some examples through all of them. So there's a very famous quote going back to Charles Box that says, all models are wrong, but some are useful. So models cannot represent everything that is going on. They're all wrong in that sense. However, some of them are useful in that we are able to still extract useful information, and they are useful to be able to inform us about some things that are going on. So all a statistical model is a mathematical representation of what is going on. So that's the system, what's really happening, and then what we observe given what is there, and it's a function of these parameters. So, in order to define what's the statistical model, we have to make some assumptions about what is going on, what's driving the system. And it's those assumptions from that is what we can then build the statistical model. So a statistical model, in another sense, is really just a set of assumptions that we are making. So it's from the assumptions, we always have to think, what are we assuming? From that, what does that mean about the mathematical representation of our system? I've already said we've got these, what's really going on the system process, and then we've got what is happening at the observation process is, explains what we see given what is there. And it's nice to split these two separate. We're typically interested in the system process. We don't care too much about the observation process, but we have to account for it in order to formally and properly account for all the uncertainties in the system. And if we ignore the observation process, we can get biased estimates. So, easiest thing to do to think about how we construct these models is to get some examples. So this is ring recovery data, and what they do with this, that in this case is a lapwings, they go to the nests, they ring all chicks that are in the nest, and they go away again. And then if an animal dies, there's an address on the ring that is on their leg that says, please send back to somewhere. And then someone sends that back to where they found the dead bird. They get the dead bird, they send the little ring back to where it says, and they record, okay, we know that the bird must have died because we found the ring, and we know when it has died. And then they just count up the number of these rings that have been returned after they've been released. So if we look at the first row here, in the first row we have 1,147 chicks have been ringed. And then 13 of their rings were recovered in the following year. 4 were recovered 2 years later, 1 3 years later, and so on. And 1,124 rings were never returned. So we don't know what happened to those birds. We don't know when they died or whether they were still alive at the end of the study. So that's the data that we have. It's very common data. They collect this on a wide number of species. So then we have to think about, okay, that's the data. What assumptions are we going to have to make, or what's happening in our system? And we typically assume that birds behave independently of each other. We can't really do too much without other information otherwise. And then come some slightly more important ones, in that if an animal dies, we are going to assume that the ring can only be recovered dead at that following time point. It can't be recovered, the ring can't be recovered 3 years later, for example. So if an animal dies, it's basically its ring is recovered immediately. That can be relaxed, but of course there's different assumptions will change the statistical model. We also assume, for example, that a bird doesn't lose its ring unless it has died. So you can't find a ring, for example, of a bird that hasn't died. Something else we may want to do is to think about, well, animals survive, so this might be annual data. So this is typically annual, 1 year to the next. We ringed the birds as chicks, but we typically know that the first year birds will have a lower survival probability than the adult birds. They're more susceptible. They're younger, they're weaker, and so on. So we might think about, well, we want the survival probabilities to be different for different ages of birds or other things along those lines. We also assume, for example, that the rings that we put on the birds don't increase their survival probability or decrease their survival probability, so the rings don't affect their survival. That's not that. There have been cases similar to this where that assumption has been spectacularly failed. There was some deer, red deer on rum, and they put collars so that when the calves were born, they put collars around the calves so they could identify them, red collars, and their mothers rejected the calves as a result. So that was a bit where they actually did affect what their behaviour was, because it meant that they weren't going to survive. So you have to think about these things carefully. So now we have a set of assumptions. We then want to build up our statistical model. When we do that, our statistical model is going to be a function of model parameters. We don't know what they are, we want to estimate them. And in our case, we have survival probability, but we've already said we want there to be a distinct first-year survival rate, which is different from adults. So we have a phi1 for the first-year survival probability, and we're assuming it's going to be dependent on time. And we have an adult survival probability, phi a, again dependent on time. But all the rings that are lost, or when an animal dies and the rings be recovered, not all of them are going to be sent back to where we want them to be sent back. They may not be found, or someone might find it but not send it back. So there's a recovery probability. So there's a probability that given that there's a ring that could be sent back, that it is actually sent back. So the probability that given the animal has died, we actually know that it has died. Typically, we're only interested in the survival probabilities. That's the system process part. We're not interested in the recovery probability. That's just the observation process part, but we need to account for it. So then, given all that, now we can write down a formal statistical model. And that is simply going to be, in our case, the probability of the observed data, given the model parameters. So what do we have here? Remember, we had number of birds released, and then we had the number that were returned later on. So we just consider each individual row in turn, and what we have something that looks like we've got some number that are released. So we've got this R1, in our case 1,147, that's the number that are released. Then what happens to them? Well, there could be lots of different things could happen to them. They could die in the first year and be recovered. So they could fall into that box. They could survive the first year. So this is just going to be die. They could survive the first year. So they would die and returned. We need to return the tag. So they could survive, then they could die, and the tag be returned. Alternatively, they could survive times 2, die, return, and so on. Alternatively, they could be not seen again. And not seen again might be they die in the first year and are not returned, or they may survive the first year, die in the second year and not return, and so on. But one of these events must happen. If an animal that is released is either returned at some point in the later year to be that it's found dead, or it's not seen again. We have to have one of these events happening. And as a result of that, we can think of that for each individual bird that is released, that it must fall into one of these categories. So it's like randomly rolling a die, for example. Suppose all of these outcomes were equally likely. If we roll a die, we know there has to get the number 1 to 6, the probability 1/6. So it's a multinomial distribution with just one observation. Similar here. This is just rolling the die, but with different numbers of bins. So rather than the 6-sided die, we've now got a lot more. And they have different probabilities of happening. So that's for every single individual. So rather than just having one individual, we have lots. We get a multinomial distribution. So it's an extension of tossing a coin r times and counting the number of heads. We toss the coin r times and count the number of times we fall in each of the different bins. So it's a multinomial distribution. So we know what distribution we're expecting the data to look like for each row, and then we just need to calculate these probabilities. And we can do that from the assumptions that we made, and in particular that if an animal dies, it must be recovered immediately, which means that if it dies, if we get the animal tag being returned 2 years later, we know it must have survived the first year and died the second year. That's the importance of that assumption. But of course, if an animal could be returned, the tag could be returned later, we change these probabilities a little bit. Everyone happy? So all with that. So each row is a multinomial, and we just product the multinomials together, and that's our distribution for all the data. So this is what all this says. So that's my managed to write down our statistical model based on the assumptions. So another example, this is the data we saw earlier, the counts with lapwings now. And they had a bit of a history. They were doing quite well, and then their numbers plummeted. So this is just an estimated number of adult female lapwings in a certain area over time. So we just get, our data is just literally at a series of t t years, how many estimated number of adult females that there are. And we only count adult females, so we don't know how many first-year birds there are. So lapwings are chicks or first years, and then they become adults, and we only recount the number of adults. But we need to know that when we set up our statistical model. So again, we have to think about the assumptions that we are going to make, because the model is dependent, or is actually constructed from what the assumptions are. Animals behave and survive independently of each other. We assume all females breed with some common fecundity rate. There's no immigration or emigration. Yeah, we can have emigration. We just don't exactly know the exact quantity of it. So we don't have new animals coming into the system, and so on. And we also assume at the observation process level that the estimate that we have is an unbiased estimator. So the expected number that we see is the true number. So then we define our statistical model. And we have to think about, we're only looking at the adult birds, but we need to take into account how many first-year birds there are as well, because, of course, first-year birds become adults in the following year. So again, we think about what are the biological processes in order to derive what's happening. So we have think about what happens from one year to the next. So the number of first-year birds there are at time t depends on how many adults they were breeding the previous year. First-year birds don't breed, so only adults breed. So it's going to be a function of the number of adults they were the time before, what their fecundity rate is, how many chicks they produce, and how many chicks survive, effectively, or the probability of chicks surviving. So rho is the fecundity rate, number of chicks being produced, phi is the probability of survival for the first years. And we assume that each individual has a number of chicks that they produce that survive is going to be Poisson with rho over 2, because this is taking into account that we assume males and equal number of males and females are born. Fecundity rate is just a total number. We're only interested in females. So it's going to be rho over 2 times phi 1. That's for each individual. They each have a number of chicks they produce is Poisson, rho over 2 times phi 1t. But they're all birds are independent of each other. We have total of NA, so we just sum of Poissons, which are the same. We just get the Poisson NA rho over t phi. It's not too much about the mechanics. It's not important. It's just how we, the idea of how we construct these things. So the number of birds we have in 1 year is basically dependent on how many a chick produced from the previous adults from the year before. However, if you think about the number of adults in year t plus 1, well, there's no immigration into the system. That was an assumption we made. So the number of adults in year t plus 1 must be related to the number of adults they were the time before that still survive, and the number of first years they were the time before that survive and then hence have become adults. Each individual survives independently of each other, and either if they were first year the year before or an adult the year before, they each have a survival probability of phi a. So each bird is simply a Bernoulli trial with probability phi a that they survive. So the total number that survive is just going to be a binomial of how many they were either first years or adults the year before. And then the observation process we assume is Poisson in this case for simplicity. Yep.
Are there other possible distributions described as 2%?
Yes. In some way. So the observation one is the simplest one. You might want to have over-dispersion here. This is not very realistic. I just put it in for simplicity. So but it's to do with the assumption that you make on the processes. So again, this is assuming, every independent Bernoulli trial, it's sensible to have a binomial. And similarly, for the Poisson, if you think of the number of chicks produced, well, Poisson's not unreasonable, but you could say, well, actually, it's over-dispersed, really. You could think that actually you could have a mixture model, that some birds aren't producing any chicks and others are, so you would have a zero inflated distribution. It's all based on the biological assumptions you are making. So it's good to know something about the system in order to build these up. Yeah? Then, if you have several models, I'll mention this later on, but so I'll jump ahead. If you have several models, then you can let the data decide which are the best models, and then that's, again, teaching you about the system, not in terms of what the model parameters are, but what actually the model is. So you can do model selection as well. Everyone happy? Right. So, some things I sort of flagged. I mean, there'll be plenty more of what decisions you have to make when constructing models. We've already had some discussions now of, well, you know, what distributions. First one is, what error distribution should we assume? That was your question. So we need to think about these things, but we determine them. We also need to think about things, what spatial scale might we want to consider, what temporal scale might we want to consider, what parameterization do we want to use, and what dependence the model parameters should have, amongst others. This is not exhaustive. So, now, so let's assume we're happy, we've done that bit, we can then construct we write down our model in terms of the distributions on each of the things that's happening. Then what we want to do is write down a likelihood function. And if we are in the discrete case like we were here, this is simply the probability of the data given the parameters. If the data we observe is continuous, then it's the joint probability density function of the data given the parameters. But given the Poisson binomial and so on, this is straightforward to write down. It's writing down the distributions, then we just look basically, if we're not sure what the mathematical representation for the probabilities are, we can just go and look those up. So these are just constructed given basically the model that we can specify. And the likelihood is very important because a lot of statistical inference is based on this. So, as a bit of discussion, I have 3 data sets here. These are all real data sets. So this is a snowshoe hare. This is a dipper. And this is to do with modern day slaves. All these data sets should look very similar. The only difference that you can detect from these data sets is that here we've got 6 years of capture-recapture. Here we've got 7. And in the last one we've got 5. So they all look the same, these data sets. Would you agree? They all look similar? There's obviously a catch coming. There is. All the different models that fitted to these are all different. Anyone want to come up with some reasons why? What might be, for example, the different assumptions going on?
The animals probably have freedom of movement, whereas the slaves probably don't.
So we could have some spatial issues going on here we might need to take into account. The last one could be animals as well, by the way. Any other issues? What other things might we want to think about? So, potentially. So, it actually, dippers is annual. Snowshoe hares was days. And this isn't even time. So, what are these capture occasions? I say, the first 2 are the same in that they are physical capture occasions. That's done on days. That's done over years. And the last one is actually, rather than going out at a series of capture occasions, they're different lists. So police records matched to hospital records. So a 0 meant you were seen by that record, and a 1 meant that you weren't. So that's one critical difference that is different in all 3 of them. What does that mean then, perhaps? Or what other issues we might want to think about? So this is 6 days. This is 7 years. That has no temporal element. That is the critical difference between these. So what, to take the 2 simpler ones. That is days, that is years. What difference might that make with regard to the data we're collecting, or the assumptions we're making? Yeah?
Miss out seasonality in the first one if you just look at everyone and the season differences.
You can't look at season in the second one either because it's always the same season.
Or it's the same.
So this could be the same month.
I mean, how can you compare the two? But that's a trend almost over a long period of time.
You've sort of the trend. So what happens to animals over years compared to days?
Well, days, you'd be looking at within their lifespan, and then over there you
Yep. Exactly. So here, days, you're just looking at a few days of what they are. They're not likely to die or move out their area. Here, if it's years, they're likely to die. So this is an open population, closed population, they can't leave. Here, it's an open population, they can die. So that's it. And then so the last one has no temporal element either, no temporal element in it at all. So I listed some things. But if you looked at the data, you couldn't tell all this apart, but each of those has different models as a result of these different issues, in particular the time element. So in the first one, when you're trapping over 6 days, you can't estimate their survival probabilities because no one's going to die. They're very unlikely to die in that time. And the reason you do it over a short time period is what you're interested in is typically the total population size. So you do it as, well, okay, it's fixed, it's closed, no one's being born, no one's dying, there's a fixed number of animals, we want to estimate how many animals there are, this is one way of doing it. In the dippers, we're not interested in the total population size typically, we're doing it 1 year to the next, we're interested in their survival probabilities. So we're interested in a completely different thing here. So we need to have that longer time scale to collect the data to allow animals to die in order to allow us to then estimate their survival probabilities. And the final one, there's no temporal order of the lists. So if you're on lists 1, take ID 1, they were seen by sources 1 and 5. We don't know whether they were seen by list 1 then list 5 or seen by list 5 then list 1. And that makes some difference. In particular, for example, you can have trap happy and trap shy if you're trapping animals. Meaning if you're seen by, if you're caught on one time occasion, you might increase or decrease being seen again because you might be having free food, so you come back to it, you're more likely to go back to the trap, or it might be a not very happy experience, you shy away from the trap. That type of thing doesn't exist if you have no temporal order. Instead, you have basically interactions, which means if you're seen by one source, you may be more likely to be seen by the other source and vice versa. But that time element of what happens first is not provided in that data in that form. We don't know what order it is. But if you need to know these questions and need to know how the data was collected in order to build the model, just seeing the data itself is not enough. That's what I'm trying to highlight here. You need to know more than just what the data set is. You need to know how was it collected. So I'll do a very quick one. So this is another data set, a spatio-temporal individual data set. So this is seals, and they tag them GPS, they let them go. So this is that's Tentsmuir up there. So this is a feeding area. So it travels out and comes back. So it basically is doing this, but it comes back that way in a slightly different, two different slightly different paths. And this is just the observations, so you can't really see it very clearly on that map. So this is just exactly the same, but with the observations. So what types of things might we need to know about this data? There's one piece of information that's missing from this, really, that is very useful to know. Anyone? Any ideas?
What those dots actually represent.
Oh, so the dot is the location of the animal at that time. What time? So yeah, so that's fine. That's longitude, latitude, but the key thing is time. I haven't said these are equally spaced in time, and actually they're not, which causes problems. Because what happens is, the little tag that you can see on the seal, it only transmits to the satellite when it's above water, and it goes underwater a lot when it travels. So it records its location roughly every 30 minutes. but it records it the next time it surfaces after the 30 minutes. So they're not equally spaced time points. So it's irregular time intervals on that. So you need to know this in order to model this, because that's very different. So you know what time is recorded, but that's what's missing from here, what time stamp is on each of those dots. So again, it's just to motivate, we need to be thinking about these things. So that's all I'm going to say about building statistical models. We'll assume now we know how the data was collected, we know the questions we're interested in, and we've built the statistical model accordingly. Then comes the next bit of fitting the model to the data to get information on what we want. Sometimes it's easy, sometimes it's not. So, how do we do this? There's two typical approaches for fitting the model to the data. There's classical or frequentist and Bayesian. So I've touched on some of this already. So in the classical sense, the classical framework, what we have, we can write down our model, we write down our likelihood, and the estimates that we can obtain of the parameters are called maximum likelihood estimators, and they are the parameter values that maximize the likelihood function. So it's an optimization problem. So we have our likelihood function, we substitute in the data values, and then we maximize that function with respect to the parameters. And typically that's called the MLEs, and then we typically have some associated error with those estimates, because we're only estimates of the parameters. We don't know them exact. That's what we want to know. So we can only estimate them because we've got the data, and then we have some error. So that's MLEs, and so we reduce it to a numerical optimization problem. And typically what we do, we go to R or MATLAB, write a function, and use the internal optimizers to do it from a statistical point of view. We're not interested in the optimization algorithm per se, we're interested in just getting the parameter values. That's one way to do it, and the sort of mindset of this is the parameters have a single fixed value, we want to estimate those parameter values. So this is actually newer than Bayesian approach. Most people think Bayesian statistics is a new idea. It actually predates this. Goes back to Fisher in the 1920s. And so we've got a single fixed value, we want to estimate it. That's one framework. The other framework is Bayesian. And the mindset is slightly different here, and the idea is that the parameters no longer have a single fixed value, but have some distribution, and we want to estimate that distribution. And sort of the idea behind this, there are some controversies with Bayesian approach, less so these days, but still, I'll mention it in a minute. The idea is we start with some initial beliefs about our parameters. So before we even go and collect the data, we have some preconceived idea of what we think the parameter values are. We then go and collect the data, and then given the data we observe, we update those prior beliefs to give us what's called our posterior beliefs. And that's what we want. That's what we're interested in, the posterior distribution of the parameters given the data that we see. So mathematically, we write it like this. We have a prior distribution that we specify. That's before we see any data, before we've even collected the data, these are our beliefs. We collect our data, and we then form the likelihood function. The likelihood function represents the information about the data. And then we combine the prior and the likelihood to give us our posterior. And we do that via Bayes' theorem. So this is where we get posterior distribution is proportional to the likelihood times the prior. Now the reason Bayesian approach was slightly controversial when it sort of started to become of interest again is because of the prior distribution. Because that's what we believe about the parameters before we've seen any data. And different people will have different priors. So this is slightly subjective. There's a subjective element to the posterior distribution. It's dependent on what the prior was. Ideally, what we typically want is what's called a data-driven posterior, where if you think we're combining the information in the data with the prior, that most of the information going into the posterior is coming from the data, the likelihood. So this swamps basically what the prior is. But there are cases where we do have prior information. It may come from an external study, and there may be very good reasons that we want to incorporate that. So it's a way of formally incorporating external information that we have external to the data that we observe. So there's pros and cons. Whereas in the classical framework, it's objective. We just have the likelihood function. Nothing external is coming in. Whether you see that as a good or bad thing is subjective. So we have a posterior distribution. We can write it down up to proportionality. So typically we can't even write it down explicitly. But it's you can't do anything with it directly. It's just too complicated. So we basically cheat. And rather than doing anything exactly on the posterior distribution, we summarize it in certain ways. I'm not going to go into the details of this, but rather than looking at this is the posterior distribution for the parameter, we will give summary statistics of the distribution. For example, the posterior mean, or a 95% credible interval, which is an interval such that 95% of the posterior distribution lies between those values. So rather than say doing it all on this high-dimensional distribution, we simplify and summarize it. And how we summarize that is something called Monte Carlo integration. Again, I don't really want to go into the details, but the idea is if we simulate from the posterior distribution, we can then use those simulated values to estimate the summary statistics. And the most common way of doing that is called Markov chain Monte Carlo. And there are some nice packages, particularly bugs and jags, for actually doing this MCMC as a black box. You just give it your prior and your likelihood, and it will spit out your summary statistics of your sampled values from the posterior distribution. There's also something called STAN. And there's also something called particle filtering, but that's probably slightly less used than MCMC. So I'm mentioning this. I don't really want to see the details. This is more for reference in future. But how you do MCMC, you start off with some initial value. And then there's different variants of it, but what's called the single update algorithm. I'll explain later why I've mentioned this. So you start off with an initial state. So for every single parameter value, set it an initial value. Then you cycle through each parameter in turn and update that parameter value. And you do this repetitively. So one iteration is you go through each parameter and update that parameter. And the second iteration you go again. So in each iteration you just update each parameter in turn, given what the other parameter values are. Don't worry about the details. So there's more there for reference later. Right, so now we're done, surely. We've got our likelihood. We can write that from our statistical model, and we've got tools for obtaining parameter estimates, either MLEs or posterior distributions. So we can estimate whatever we want, given the estimate the parameters in our model. Unfortunately, things crop up that go a little bit wrong, in that it's not quite as easy to do this in certain cases. There's other questions we want to answer. So I've just picked up on two issues that I've addressed briefly. One of them is, we've already mentioned earlier, model choice. So are survival probabilities time dependent or not? And so on. What dependence do we have on the model parameters? What error structures do we have? So that's one issue. And the second issue is what happens if we can't write down the likelihood? I've sort of implied that you could always write this down, but you can't. So, very briefly, let's address both of those issues and things we might want to think about. So, mention model choice. Let's think about, go back to our ring recovery, if you like. Are the survival probabilities time dependent or not? Or are they a function of the harshness of the winter? Most deaths for birds happen in winter. We want to be able to discriminate between those. So what do we typically do? Well, first of all, we define what are our set of plausible models. What model dependencies do we believe might be present? We can list them down. And then for each of those parameter dependencies, that's a statistical model. We can fit the model to the data and estimate the parameters. So we can do that, and we can estimate, given a model, we can estimate the parameters. However, we want to be able to discriminate between those different models, because actually being able to say that a parameter is time dependent or not is also telling us about the system. We're telling us time that survival changes significantly from one year to the next is very different information than the survival rate's the same every single year. So it tells us something about the system. So we're able to discriminate between these different models. And again, you can do that in the classical or the Bayesian framework. And typically what you do when you have model choice, not only do you present the results of just, if you just have one model, you present the parameters, the estimates of the parameters. When you have model choice, you have sort of a two-step process. You have what are the top models. I'll describe how you can discriminate between the models in a minute. And then given the models, what are the parameter values? But the additional thing you can do is to obtain modelled averaged estimates of the parameters that take into account not only the parameter uncertainty, but the model uncertainty. Again, flagging it, and you'll see it in a minute, just to give you an idea of you can do this modelled averaged estimates. So, classical model choice, several different ways of doing this. I've really picked up on one which is useful. It's a Keiki's information criteria, typically called AIC. And so for each model that you have, or each parameter set of parameter dependencies, you can calculate the AIC statistic. And it's two parts to it. First part tells you how well does the model fit the data. And the second part, so this is minus twice the log likelihood evaluated at the MLE, it's called the deviance. And the second part is how complex is the model. So PM is the number of parameters in your model. So the first part tells you something about model fit, and the second part tells you about model complexity. You want the best fitting model for the smallest amount of complexity. So it's a trade-off between those two things. If you make the model more complex, then it has to fit the model much better in order to make it worthwhile having that additional complexity. And the model with the smallest AIC is deemed to be best. And you can also assign weights that allow you to do this model average estimating. So that's classical. I've just mentioned one. There's plenty of other classical model selection tools. And the Bayesian framework is quite interesting, and this was basically most of my PhD was involved in this. And you think of, before we had a posterior distribution of the parameters given the data. Now we're going to extend our definition of parameters in that sense. So we're going to think of the model to be a discrete parameter to estimate. So we might have 3 different models, and we treat of those models as parameters. So then we turn the Bayesian handle. Again, we form the joint posterior distribution over all our unknown things, which are the model parameters and the model, given the data. And you can just write it down. You basically get the likelihood of the data given the parameters in the model. You have the prior for the parameters given the model, and you have a prior probability for each model. And now, again, you can sample from this posterior distribution, and then that will allow you to estimate quantities of interest, which include the posterior probability of each model. The MCMC is a bit more complicated here, so I'm not mentioning it at all, just to flag that it is more complicated, but you can do it. So what are you interested in? I said particularly what you're interested in is your posterior probability for each model. And then you have posterior distribution for each model, and again then you can have a posterior modelled averaged estimate for your parameters. So you get, and this is simply a weighted average of your posterior distribution for the parameters given the model, weighted by the corresponding posterior model probabilities. So it's simply forward weighted average. So you can do that if that is of interest. So, give you an example. So these are our dippers again. This was capture-recapture data. No, there was no recoveries. So we have different models here which corresponds to the dependence. So here we've got a constant survival and a constant recapture. Here we've got a time-dependent survival, constant capture, so on. And here we've got 2 survival probabilities dependent on whether it's a flood year or non-flood year. Just some years are one phi, some years it's the other phi, and constant p. So we calculate the AIC, and you can see that is the best model. And you can also do this in the Bayesian framework. You calculate posterior probabilities, and it actually, again, we get this model is sort of identified as best. It doesn't always mean that the classical and the Bayesian model choices will give you the same output. You can get it. The AIC might favour one model. Model with the highest posterior probability might be a different one. There's no reason that they should match. You can do much more. So this was a fairly simple one. This is a Soe sheep. And you can see actually there it's a ring, a tag on its year, how you identify them. So here we had 3, 4, 5, 6, 7, 8 different possible dependencies that the survival probabilities could be a function of, or covariates, to do with different attributes of this sheep or environmental conditions. So the survival probability could have been a function of up to 8, and we had some some other things as well. And what we have in the plot is the posterior probability that that covariate was important, or the survival probability was a function of that covariate for different ages of the sheep. So just to highlight, you can get much more complicated structures going on, and so on. So that's model choice. Now we have some tools, but what happens if we can't write down the likelihood? There's two reasons this may happen. One is that we might only be able to write down the likelihood as an integral, which we can't actually do. Or it might be that the likelihood is possibly a summation, but the summation is too big that feasibly we can't calculate it. Then we've stalled at the moment because everything I've talked about now involves being able to explicitly evaluate the likelihood function, either to calculate the MLEs or to calculate the posterior distribution. So just to give you why does this happen in practice, let's look at the dippers again. So again, this is nice and simple. We release them and then we see them alive again, but it's very, very similar to this structure. And we get our likelihood function. Again, I'm not worried too much about how we get it. But this is our function. So it's simply a function of phi, survival probability, and p, capture probabilities. And this chi term at the end is still a function of these p's and phi's. Don't worry about the function, what it is, just it looks of that form. So that's fine to some extent. The difficulty comes is actually we want to write that the survival probabilities are different for each animal. And in particular, we have what's called a random effect model. So we might write the survival probability in this way. So we have that they're time dependent plus some individual effect, where the individual effect comes from some distribution. And then when we write down the likelihood, the parameters in our model now are actually the capture probabilities, it's the alpha, and it's the sigma squared. And we integrate out the epsilons. So this is what this expression is here, but we can't do this integration. So it's analytically intractable. So second example where this arises, I'll get back to our lapwing data again with the counts. We've seen this model before. This is just a reminder of what the model is. And then we can write down our likelihood as a summation over all these true values that we don't know. Because what we want is a function of the data given the parameters. The parameters are the fecundity rate and the survival probability. But we have all these unknown true population sizes that's driving the system, we have to sum out. So we get a summation that we can't calculate. So again, we can't write down the likelihood. So this is sort of an area of really still ongoing work, more generally within the statistical community, of what do you do. There are some techniques, and how well they work depends on how complicated your situation is. If they're low dimensional, then they generally work fairly well. So I've said we have an integral. Let's go back to there. We have an integral. But if you notice, this is a product over, this is just one dimension. So we've got one-dimensional integral, which means we can use simple mathematical tools to do a numerical integration approximation to that integral. Think of like trapezium rule. If you just got to try and do an area under a curve, we could do a trapezium rule. We can do a similar thing here. It's only one dimension. So we could use something like Gaussian quadrature. In the Bayesian framework, we can do something a bit slightly different. Again, if you think about when we had model uncertainty, we extended the posterior distribution to include the model to be an unknown parameter. Well, here we can treat the true underlying population sizes to be unknown parameters and calculate the joint posterior distribution over all those, and then we can marginalize to get the posterior distribution of just the parameters. So this is our first capture-recapture one. Oh, I didn't need to go backwards, it was on one. So again, this is our integration. We can't do it, but we could just, the product of one-dimensional integrals. So we can do basically approximation, Gaussian quadrature, or if you think simple case, trapezium rule. Alternatively, we could do Bayesian data augmentation. Like I said, we treat the unknown parameters, the things that we're trying to integrate out, which in our case is the epsilons, as parameters, and we form the joint posterior distribution over the data and the parameters. And then we can write all this down. We do a MCMC to sample from this joint posterior distribution. And what we really want is the marginal distribution. We want to integrate out the epsilon. But actually, if you have an MCMC, this is done automatically. So your MCMC gives you a set of sampled values for x and e for each iteration that you run your MCMC algorithm. If you're only interested in, so these are the wrong way round. It should say thetas and x are all the wrong way round here. Yeah, so those are the wrong way round. That's just the posterior distribution of theta given x. You integrate out the epsilons, which means you have in your MCMC output, you have at each iteration, you have a set of values for your thetas and your epsilons. If you want the posterior distribution of just the thetas, you just ignore what the epsilon values are. So it's really that simple. It falls out automatically. So that was first one, count data, same type of thing. If you're classical, you can do some approximations. If you're Bayesian, you can do data augmentation. So how many of you know about MCMC? 2, 3. Right, so I did this, right? So for those of you that know MCMC, what I want in my next slide is a plot that looks like this. So I'm going to do what's called, for those of you that know, it's called an ACF. Right, I want one that looks like that. Right, one spike and then virtually nothing. What I get, if you do this using the single update algorithm I described earlier, you get something that looks like that. Again, notice the lag. You want this sort of to look around about lag 10. It's horrendous. It's very, very poor. So although you can do these things, they might not work very well. But that bit aside, if you can then get these things to work, or at least you run it for a very, very long time, you can get some very cool output. So this is again for the lapwings. Remember you had your estimates of population size over time. The type of output you get is this is your estimate of your adult population size plus your 95% uncertainty interval. We also get your first years. We didn't see any first years at all, if you remember. We only saw adults. So from just the adult data alone, we can still get estimates of the first years. Now, you might not be interested, you might just be interested in the model parameters, but actually in some of these situations, you are interested in what the true underlying process is, as well as the model parameters. And also, what we have on this side is basically a function of the left-hand side. So there's something called a red data list, where they look at changing population size of animals over a 25-year period. And you can have a green, amber, or red flag, and one of the things that they look at for this list, red is bad for conservation, is the change in population size over 25 years. So it's red if the population size is halved or more. It's amber if it's between 0.5 and 0.75 compared to 25 years ago, and above 0.75 it's sort of green. It's only one of the indicators, this. So it looks at the population size now compared to 25 years ago. And if you just use the estimated values, your data points, you would get the black line. And you can see you'd go from green to red. So from 1 year, you'd go from green to red, which is just doesn't make any real sense. You wouldn't change that population size that quickly. And it's to do with the fact that there was a lot of error in your observed counts. However, if you estimate these, you do it in this analysis, then you get what the blue one is. Not only do you get sort of a much smoother change in this 25-year statistic, you also get error bars of what actually, so these are 95% credible bars, so 95% probability the parameter lies within the upper and lower bound. So you can pull out very interesting statistics as well. So it's just to give you a flavour of some of the things that you can do. So that's sort of a whistle-stop tour of some applications, some things that can arise within the environment and within the particularly ecological side of things. So just to emphasise, two processes to think about. One is how you to build, given the data and the data collection process, how you build a statistical model. And you need to think about the assumptions that you have made and the data collection process in order to build it. And it's useful to split into two parts. One is what's really going on in the system process, what's really happening, and what's driving those changes over time if it's a temporal system. And then given that what's really happening at ground level, what are you observing given what's truly there? So split out the processes. Just to flag, I say you have these assumptions. Sometimes assumptions are made for mathematical convenience, not biological realism, in order typically to simplify the model specification and probably even more so to simplify the model fitting process. So you do need to be realistic. When developing statistical models, you could develop a statistical model, there's no way you can fit it to the data. That's not going to help anyone, so you need to try and be sensible, but you also need to be aware if you are starting to make an assumption that's not biologically sensible. So there is a bit of tension there. And even though you can do all this, you write down your model, just to flag, model fitting is not necessarily non-trivial. And a lot of work is in actually getting this methodology, sort of standardized methodology to allow us to do this, apply this to all different systems. So yeah, any questions? I'll stop.
You talked about AIC. What are the assumptions with AIC, if there are any?
Well, there's no sort of assumptions with the AIC, it's just how it's derived, and it's to do with the Kullback-Leibler distance, to do with your information lost. There's not the only ones. There's Bayesian information criterion, called the BIC. There's also variants of the AIC, corrected and quasi versions, and it's all to do, one of them is to do with small sample size. In terms of ecological models and AIC, there's a very nice book by Burnham and Anderson that's very interesting, but it's particularly applied to ecological type models. So that probably doesn't answer your question, but it's not necessarily assumptions that you're making. It's just a way of discriminating between different models, but the way that the actual formula has arisen is to do with how the behaviour in this, I said this Kullback-Leibler distance to information loss. But there are many others.
Maybe a simple one. Is the maximum likelihood function the same as the likelihood function in Bayesian method?
So the likelihood function is the same.
Okay.
So the likelihood function you maximize in the classical framework is the same as the likelihood function you combine with your prior in the Bayesian framework. It's exactly the same form, yes.
Okay, I see.
You're just doing different things to it.
Yeah, one is to get one value, the other is get a distribution.
Sorry?
One is to get one value.
One is to?
Get one value, get one parameter, get one estimation.
It's to do, so it's a philosophical argument here as well. I'm a pragmatic statistician, so I will use what I think is the most appropriate approach for the question being asked. In practice, what this has happened over the years has meant with, I have a likelihood that is analytically intractable, I can't fit it classically, hence I'll be pragmatic and be a Bayesian because I can fit it in the Bayesian framework. But there are philosophical arguments with the classical framework. You assume that the parameters are a single fixed value and you want to estimate those single fixed values. In the Bayesian framework, you start with the philosophical argument that you have a distribution for the parameter. No more questions. I've got one more thing to say to you all, which is good luck and enjoy your studies. If you ever want to chat to me, I'm around in the ATR, at the Turing once a month approximately, but I'm also in email. But it'd be nice to chat to any of you if any of you are interested. But anything, not just ecology and environment. Just, tell me what you're doing. I'd be interested to know.